A very good afternoon, everyone. I am Dr. Manas Patimbatra, Assistant Professor, Faculty of Pharmaceutical Sciences at London University. Welcome to the third and the fifth session of the FDP, titled Paradigm Shift in Pharmaceutical Education and Research in Post-Pandemic Era, organized by Faculty of Pharmaceutical Sciences at London University. So today the session will be carried out by Dr. Mohammad Sarwar Hussain, sir, Associate Professor, Deputy International University, Dhaka, Bangladesh. Let me share a few moments of his professional glory. Dr. Mohammad Sarwar Hussain, sir, completed his BPharm and MPharm program from the Department of Pharmacy, University of Rajshahi, Bangladesh, with his distinction results. Currently, he is serving as a Associate Professor in the Department of Pharmacy, Faculty of Allied Health Sciences, Deputy International University. In addition, he also worked as an Associate Head, Department of Pharmacy, Deputy International University since May 2019 to April 2021. Dr. Hussain has been awarded his PhD degree from the University of Tsukuba, Japan, in the year 2016. During his PhD research work, he was awarded the very prestigious Young Investigator Award in 2016 from the University of Tsukuba, Japan, for his outstanding research work. He was also awarded the Impressive Teaching Excellence Award in 2020 from Deputy International University, Dhaka, Bangladesh. In addition, he was awarded a gold medal for academic success and got numerous scholarships throughout his student life. It is to mention here that Dr. Hussain has an experience in teaching and research for around 14 years. During his time, he has published several research articles in different renowned journals. He also involved in collaborative research work with different national and international universities. He always pays attention to modern technology-based education and research for the student to meet global demand. Besides his sincere academic duties, he is also involved in different co-curricular activities. His teaching philosophy is aligned with effective mentoring to help the students for their self-development, acquire quality knowledge and academic and successful research, increase positive emotion for sharing knowledge with others, and influence to improve their innovative thinking to reach career goals. So his motto implies is always be mentored and always be mentored. So today he will be delivering a talk on the session entitled Novel Dominant Scheme Identified Mutations under SIM1 and MC4 are genes in obese pedigrees. Before starting the session, I would like to request the participants that if they have any queries, they can post their queries in the chat box, or else if they want one-to-one conversation with the speaker, then they can switch on their camera and also they can unmute them and they can have their queries to the respective speaker. So with this, I would like to request Dr. Hussain sir to start his session. Sir, the stage is yours. Thank you, sir.
Thank you, sir. Am I audible clearly?
Sir, you are audible clearly, sir.
Okay, thank you, sir, for giving me the opportunity. So, this is a very good afternoon to you all. At first, I convey my veneration to the Department of Pharmaceutical Science, Assam Downtown University. I'm very much grateful to you all and all the participants who are giving time here today. And it is an immense pleasure for me to present myself before you. So thank you all for time here today. I am starting my presentation. So is it visible, my screen?
Sir, your screen is visible.
Okay, sir, thank you. So today's title, the Novel Dominant Screening Scheme, Identified Mutation of the SIM1 and MC4 gene in pedigrees. So already Dr. Manas sir introduced myself before you. So I am not introducing again myself, but I am informing you this is the part of my PhD research work which I'm going to present before you. So at first, I'm going to start with the background of my study. In order to identify gene regulating sleep wakefulness, our lab performed a large-scale forward genetic analysis of randomly mutagenized mice to the sleep abnormalities. And the mutation was introduced by injection of ethyl nitrous urea to the B6J altered male mice. And this genetic analysis was performed through the dominant screening of forward genetics. And in parallel with the screening of sleep abnormalities, our lab found some mice which are exhibiting obvious obesity, and we hypothesized that these mice have been dominantly inherited by the mutation of obesogenic gene, and possible to identify the gene of causing obesity through the dominant screening of forward genetics. So this was the starting point of my research. So as you know, the obesity was imbalance between food intake and energy expenditure. A lot of diseases like diabetes, heart disease, cancer, hypertension, and many other complications are associated with obesity. And we also know there's a lot of factors of causing obesity, and genetics is one of them. And in my research project, the part of genetics is important for us to do my project work. We also know that after the seminal discovery of leptin and long form of leptin receptor, the hypothalamus region of the brain, it has been indicated as an important region of the brain for food intake and energy expenditure. And thereafter, some other genes have been identified which are involved in the food intake and energy expenditure. They are located in the region of hypothalamus. And if we see the previous result, through the past forward genetic studies on obesity mice, some genes leptin and leptin receptor mutation have been identified through spontaneous obese mutation. And some other genes, cycleptin gene, leptin receptor, and MC4R mutation gene, have been identified through the recessive screening of immunomutagenized mice. But there is no successful dominant screening has been reported for the identifying of gene causing obesity. So what is the possible reason of the no success? Actually, the different metabolic effect of different quantitative trait locus, that is KTL, between the mutagenized mice and backcross mice, because these mice have a lot of polymorphism in the genetic area, genetic locus, and different metabolic characteristics. And these differences confound the mutant locus, and that is difficult to identify the QTL through the statistical analysis. And for that reason, clear phenotype was difficult to identify. So the further processing of identifying gene through the forward genetics was difficult. And another point in case of obese mice, that obese mice is unable to produce progeny through the natural mating. So I have taken to work on the studies through a process. One, using of C57BL6J and C57BL6N mice for crossing, because both of the mice have almost same genetic background, same metabolic characteristics, and the less number of polymorphism in the genetic region. As a result, it is very much easy to identify the mutant phenotype after backcrossing, and easy to identify the linkage analysis and result for the further processing. And another point was routine use of in vitro fertilization. With a very short time, we can produce a lot number of mice by in vitro fertilization process. So by using these two process, we have started my research strategy. And the purpose of the study was to evaluate the efficiency and reproducibility of our new forward genetic scheme of dominant manner, and to identify a gene mutation causing obesity through the dominant inheritance. And if we find the new gene, that would be a gene which have been identified through the dominant manner of forward genetics, maintaining the food and energy intake and expenditure. So the strategy of identification of mutation of obese pedigree. In our screening strategy, we have been injected ethyl nitrosourea at the age of 8 weeks of B6J wild type male mice, that is G0 mice, and it has been subjected for mating with the B6N wild type female mice through IVF and produce F1 mice. Among the F1 mice, some exhibiting obvious obesity. So this F1 mice is then again crossed with B6N wild type female mice through the IVF to produce N2 mice. It has been dominantly inherited, and this N2 mice has been subjected for linkage analysis to identify the quantitative trait locus, that means KTLs, and then subjected for the whole exome sequencing to identify the positive gene in the linkage area, which have been responsible for producing the phenotype. And finally, we perform the phenotype assay related to the identified gene to confirm the function of the gene mutation. As there is no selected criteria, because it was the first methodology for dominant screening of obesity, for that reason, we didn't know what type of age and what was the body weight for the selecting of obese F1 mice. For that reason, we have selected the heaviest obese mice from the different ages. 5 obese mice at 18 weeks, 1 obese mice 10 weeks, 5 obese 28 weeks, and 2 obese mice at 32 weeks. It has been randomly selected among the screening of more than 500 mice. Actually, we have selected the 13 heaviest obese mice. And this is the body weight histogram of these obese mice. Here, the number of mice near about 2,900. And here we found the 2 mice exhibiting highest body weight more than 50 grams at the age of 18 weeks. So we have selected the heaviest 2 obese mice for my further works and named them obese 10 and obese 13. So now for obese 10, through the linkage analysis, we found the associated locus of obesity phenotype on chromosome 18. And also high LOD score through the linkage analysis as per the result of statistical analysis, high LOD score at chromosome. So we think that the mutant locus of the genetic mutation has been found in the chromosome 18. So further analysis was performed by using large number of mice only for chromosome 18, and we found high LOD score. Here the number of mice 206. So which exhibit and confirmed our hypothesis that the mutation, genetic mutation is located in chromosome 18. Then through the whole exome sequencing, yes, we found the mutation of MC4R gene under the peak of the high QTL analysis, high LOD score. And we also know that through past research regarding MC4R gene, the obesity is correlated with the man and mice. So this dominant screening strategy for the obesity of MC4R gene causing obesity, and who is also confirmed through the previous study. So our screening is okay to find out the mice which have been obvious obesity of causing the mutation in MC4R gene. Okay, then we go for the single nucleotide analysis for the screening of obesity mice in case of MC4R gene. And we found that the single nucleotide substitution, that is thymine to adenine, is found results in only obese mice, but there is no change in the non-obese mice. That means the single point mutation have been done only for obese pedigrees. And this transversion of this nucleotide thymine to adenine substitution results the change of amino acid from tyrosine to the stop codon. And this mutation is located at the beginning of the first transmembrane domain of MC4R protein. Next, for obese 13, according to the obese team, we also performed the linkage analysis, and the associated locus of obesity phenotype was found in the chromosome 10. Here is the area, chromosome 10. And through the linkage analysis, we also found high LOD score, that means association or segregation score, at 10, chromosome 10, that is 7.9. So you have to synthesize that the mutation is located at chromosome 10. So further analysis was performed with large number of mice only for chromosome 10, and we find a very high LOD score. So initially, the linkage analysis confirmed that mutation of responsible gene is located in this area. Then whole exome sequencing found the mutation of Sim1 gene. And through the previous strategy and research, we also know that the mutation of Sim1 gene causing obesity was found both in human and also in mice. So further analysis of the previous result and whole exome sequencing again proved that this strategy confirmed to identify mutation regarding the obesity through this dominant manner inheritance of forward genetic analysis. Again, the sequence analysis only for the Sim1 gene, we found the nucleotide sense, single nucleotide sense, only in the obese mice, but there is no sense in non-obese mice. And here also the nucleotide sense from thymine to adenine. And this substitution results the change of amino acid from methionine to lysine. And this mutation is located in exon 4 of Sim1 gene. As you know, the Sim1 is a basic helix to helix fast transcription factor, and is the well conserved in all living organisms. And the mutation was found in past domain of Sim1 protein. The Sim1 protein highly expressed in paraventricular nucleus of hypothalamus, and it's very much important for the development of hypothalamic region, paraventricular nucleus. And Sim1 acts as a transcription factor by dimerization with ARNT2 gene. And by the dimerization of ARNT2 gene, Sim1 shows its high transcriptional activity. So after the mutation, what's about the result of its transcriptional activity? So we confirmed the Sim1 mutations after mutation its activity through the luciferase assay. So in case of luciferase assay, we designed SIM1 wild type protein, SIM1 mutant protein, and ARN2. As you know, the SIM1 and ARN2 act as a transcription factor by dimerization. So singly SIM1 protein didn't show any transcriptional activity. Only SIM1 mutant, no transcriptional activity. But SIM1 and ARN2, the dimer. But interestingly, here we found that SIM1 mutant, that means the mutation showed loss of transcription. Again, in case of dose-dependent manner, we found that with the increase of amount of wild-type SIM1 protein, its transcriptional activities increases, but there is no result in case of SIM1 mutant protein. So further analysis also confirmed that this mutation showed the loss of transcriptional activity of the SIM1 protein, and the mutation of SIM1 protein is correlated with the obesity because lackings of development of parameter nucleus, which affect the other gene which is responsible for energy expenditure and food intake, so promotes the obesity. So this phenotypic analysis also performed our methodology, that is efficiency and reproducibility. So the short summary of my initial part of this research, we have established 2 obese pedigree through the dominant screening of obesity using C57BL6 substrains. In case of obese 10, a single nucleotide substitution of MC4R gene found only in obese mice, and this substitution results in a premature stop codon, and tyrosine to stop codon located in the first transmembrane domain of MC4R protein. And in case of obese 13, here we found the single nucleotide substitution in SIM1 gene. This is also only in obese mice, and also this substitution results in amino acid chains in a passé domain which lacks the transcriptional activity of SIM1 protein. Now, for again the confirmation, the metabolic phenotype, the effect of the mutation. So in case of metabolic phenotypes of MC4R mice, mutant mice, and SIM1 mutant mice, here we found that in both cases, the mutant mice exhibited high body weight, high food intake, high epididymal fat, high leptin level, high insulin level. Both SIM1 mutant and MC4R mutant mice. But there is no significant increase of glucose level, though slightly high, but there is a significance. So this may be the secondary effect of obesity or high insulin level, which may compound the increased level of glucose. Further phenotypic analysis was performed through the hypothalamic gene expression in obese mutant. So in case of the study of mutant pedigrees, we also produce another group of obese by diet-induced. So this diet-induced obesity of mice show the high body weight, and we use high diet fat, here 60% fat, and another group we use low diet fat, that is 30% fat, and other is here. So after collection of the tissue from the brain, the quantitation of hypothalamic neuropeptides gene was expression. We found the low arginine vasopressin level in all, and AGRP, agouti-related peptide gene expression, is significantly decreased than wild type SIM1, MC4R, and diet-induced obese mice, so diet fat here. So this result is correlated with the previous result of diet-induced or similar mutant or MC1 mutant decrease. That the common decrease in adrenaline vasopressin and AGLP mRNA is due to the secondary effect of obesity associated with increased leptin and insulin level. Decreased adrenaline vasopressin, agouti-related peptide mRNA consists with heterozygous SIM1 mutant mice that also have been found in the SIM1 mutant mice in the previous reported journal. And the decreased HRP mRNA in diet-induced mice also consistent with the previous report. Next, the quantitation of hypothalamic neuropeptides regarding the oxytocin, thyroid stimulating arrays mRNA consistent with the heterozygous SIM1 mutant mice. This has been reported, and decreased orexin by detail 2014 also reported. So this hypothalamic gene expression result also further confirmed our new methodology, reproducibility and efficiency, that our methodology is working well and produce 2 mutant obese pedigree, which confirmed the efficiency through the phenotypic assay and hypothalamic neuropeptide genes expression also. So, to the conclusion, this was the first successful dominant screening of forward genetic study for obesity by the combination of using B6 substrate, including use of in vitro fertilization and whole exome sequence. And both of the mutations showed strong correlation with the obesity. The study further confirmed the major role of MC4R and SIM1 gene mutation causing obesity through the phenotypic assay. And the study exhibited distinct gene expression pattern for MC4R and SIM1 gene mutation in the hypothalamus, which has been confirmed through the hypothalamic gene expression assay. So the future direction from my study, this dominant screening strategy using B6 substrate is a very much applicable method in obesity screening. Also, any other research area, if we want to identify any kinds of candidate gene that affect the variety in behavioral or metabolic phenotypes or any other disease causing the mutation of gene. Actually, if we use this strategy by using the B6 substrate, IVF, or whole exome sequencing, or natural mating and whole exome sequencing, all are applicable to identify any candidate gene mutation. So thank you all for your time here today.
Thank you, Hossein sir, for your informative and interesting session. So I think the participants are benefited with your whatever you have shared with the participants. So this session is open for questioning now. So any participants wanted to have any interaction with the respected speaker, questions in the message box, or they can directly talk to our speaker, sir. So meanwhile, sir, I also used to work on this particular field, although I'm not into much genetics. I basically work on the field of pharmacological evaluation of some natural product like constituents. So I have two queries actually. First query is regarding the animal models. So according to you, which like animal model is suitable for obesity? Because in various papers, there has been reported that steroid are also used, as well as albino mice, C57BL6J mice. So according to you, which one is more suitable for anti-obesity studies?
Thank you, sir. So I am giving you the answer. Actually, here we show. Can you see the slide? Hello, sir. Sir, do
Yes, sir, you are.
use B6J, DBA2, C33, and other mice, DBA2, BC3, AC, as any other.
Okay.
All of the mice have almost different polymorphism, lot of polymorphism in the genetic area, more than 5 million that has been identified.
Okay, sir.
So if the polymorphism is more and more, this is difficult to identify the QTL for the first linkage analysis. So if we cannot identify the actual position through the QTL analysis, so we cannot identify the exact location in the chromosome which gene is responsible. So further processing of whole exome sequencing will be difficult. But B6J and B6N mice both are almost same genetic characteristics. Only 70,000 polymorphism is present in their genetics. As a result, if we produce the new progeny, this is very much easy to identify the phenotype. If I show you another slide here from my backup slide, you can easily identify it. So look at this. This is the C57BL6A and C57BL6N. So just this is their characteristics. So wild type and mutant type, when we crossed, or that means with one another and produce the N2 mice, there's significant difference of mutant type and wild type. So we can easily identify it, the phenotype from the wild type if we make two groups, wild type group and mutant group. But other mice, as they have a lot of characteristics, so many other characteristics and many different details which compound. So first one, difficult to identify the exact quantitative locus where is the gene mutation is present. Another one, due to the presence of different characteristics with the same area and same manner. So this is very much difficult to differentiate between the wild type and mutant type. Actually, that was the main reason by using the CP7, BL6J and BL6 mice rather than other mice. So you talked about other mice using in Indian area in Bangladesh.
Exactly.
But I don't have any experience about the mice. What was the genetic characteristics and the polymorphism, number of polymorphism. If it is too much high, that should be difficult to identify.
So actually, sir, most of the time we seen that, sir, to develop a hybrid diet model, so it takes time, sir.
Yes.
And if we use Swiss albino mice, or mostly, sir, Western rats are used, they
That
don't become obese, sir, but their lipid profile either it becomes bad, sir. So, the HDL will decrease, triglyceride will increase, cholesterol will increase, but they don't become fat. But when C57 BL6 mice used, and they were fed with high fat diet, sir, so they become obese, sir. And that is a significant obesity, sir,
yes.
as compared to an animal who have taken some control.
Yes.
Yes, sir.
So
And-
yes, And in case of other mice and C57 BL6 mice, so I showed here at the 13 obese pedigree. So all the obese pedigree actually not by B6A and B6N. The last 4 pedigree was B6 and B6N, which show the highest body weight. But others, B6J, DBA2, B6J, BL6, and other groups, but we did not find actually the high body and low body weight group. There is no significant difference. The mixer, so we cannot differentiate the two groups, which group is for high body weight and which group is for low body weight. This is very much this was very much difficult to differentiate between the two groups.
Okay.
But in case of differentiate, there is a high difference. The high body weight group more than 50. At the same time, low body weight group, we can easily differentiate the both of the groups. So according to B6 and substrain, that should be a good choice.
So one more query from my side, sir.
Yes, sir.
So you have basically MC4 gene as well as POMC, pro-PO melanocortin.
Yes.
And basically, what if the level of this MC4 gene or POMC gene increases, elevated, sir? And adiponectin, it's a anti-inflammatory adipokine, sir, adiponectin. Adiponectin is reported to develop MC4 gene as well as POMC. Okay. Now my query is that, sir, is the report of any, sir, any agent or any compound that in the clinical trial that targets the POMC or MC4 genes?
Thank you, sir. Actually, that was not as a part of my research because we observed the reproducibility and sensitivity of my methodology. As we found the SIM1 M136K gene, just we correlate and compare the result of some phenotypic analysis and also hypothalamic expression. So what's the result? Is it similar to the previous result or not? If it is similar, that means the MC4 gene mutation as it caused obesity. So yes, that was the ending of my research. But according to your question, MC4 gene actually regulate the food intake and energy expenditure.
Exactly.
If it is the normal gene or wild type gene, but when the mutation of MC4 gene, it cannot work. Actually, MC4 gene, you know, it activated by melanocortin stimulating hormone, and also the melanocortin stimulating hormone comes from pro-melanocortin stimulating hormone, and pro-melanocortin stimulating hormone also activated by the action of a leptin gene.
Yes.
So when any kinds of mutation, either leptin or melanocortin or MC4 gene, so if any part of the area is mutated, so the MC4 gene or the brain actually doesn't get the right message regarding the presence of fat in our body. So brain thinks we don't have enough food, so we need food more and more.
Exactly.
Then we take food, or mice take food, and they become obese because they are gathering a lot of fat in their body due to the wrong response or no response by leptin gene or melanocortin stimulating hormone or MC4 gene. So actually MC4 gene it's okay, then it regulates the body weight in the normal manner, food intake and energy expenditure, that is homeostasis. But if any mutation, in that case, they didn't get any actual response. So they think we need more and more food. And by taking more and more food, gathering the fat and becomes obese.
Yes, sir. There will be high energy intake and less energy expenditure.
Yes, sir.
Yes. Sir, anyway, sir, thank you for your sir, wonderful session, sir. We have
Thank you, sir.
not received any query from the participants. Sir, so I think we can wind up this session, sir. Thank you for your wonderful, interesting, and innovative session. I hope that the participants have increased their knowledge on this particular topic, that is obesity and related genes, which participate in the pathology of genes. So with this, I would like to end up this session. So thank you for participating in this faculty development project. Thank you, sir.
Thank you, sir. Thank you very much, and also thanks to all of the participants. And this is my immense pleasure that they are present here and they are another for questions hearing. So I am very much happy to share my experience and knowledge before you. So thank you.
Thank you, sir. Thank you so much, sir. Thank you. So I would like to request the participants to fill up the feedback form. Activate for 2 minutes only. So I'd like to request all the